All right, then we start, inshallah. All right, today our discussion will be about the pain. And as we have discussed before, the pain is sensed through nociceptors. These are the pain receptors. Okay, and a number of chemicals. A number of chemicals are involved, which can stimulate these pain receptors. So we'll just discuss those. And also we'll have some discussion about the various types of pain, broadly speaking, fast and slow pain, and which types of nerve fibers they transmit the pain signal. Okay, and we will also see how pain can be suppressed, that is analgesia. So, what is pain? You must remember from your last semester discussion that it is an unpleasant sensory and emotional experience that is associated with actual or potential tissue damage or sensation of physical or mental suffering. So, basically, it is tissue damage or potential tissue damage. Special quality is that it is very subjective. Only the person who is experiencing the pain can describe it. Same degree of injury could be there, but one person feels a lot of pain, the other person does not feel that much pain, rather disregarding that pain at all. But the most important aspect of pain is that it is protective in nature, because it provides a warning signal, that something is not well. There is something wrong going on in the body and something must be done. So it is protective in nature. So, as I mentioned before, pain receptors are called nociceptors. And one very important quality of these receptors is that these receptors do not adapt. By adaptation means that if the stimulus remains there, the signal does not go down, it does not disappear. For as long as the signal is present, the response of the nociceptors remains there. Okay. Now we will discuss the different phases of nociception. These are very similar to whatever happens in other sensations. So the first thing that happens is transduction. As I mentioned before, every sensory receptor is a transducer. That is, it converts one form of energy into another. And for all practical reasons in the body, it is converted into electrical energy, electrical signals. So this is the process of sort of excitation of nociceptors. Second phase, of course, would be then be transmission, sending these signals towards the central nervous system.
Doctor,
So, yes, please.
are you switching the slides or the still the first slide?
Yes, I am switching, but it is not being switched there.
Now
Let
it's
me
switching. Oh,
let me see what is happening. Share screen, window. Now it is there.
yes.
But there is a little problem when I switch it on to slideshow. There is a so we can probably just use this one. Slides are clear.
Are
Okay.
you changing?
Now, do you see the slide number 4 or no?
It's 5 now. If
Okay.
you want to put the slide maker or the show, I think you should turn it on first, then turn the share screen.
Okay. So I will
Just share
stop
the
sharing.
entire screen.
Huh?
Below.
Okay. Are you able to see the screen now?
We are seeing the first slide.
Yeah. Now the second.
No,
It's not coming second.
no, doctor.
Mhm. There is something wrong with that. So
If
Doctor,
yes.
don't use the full screen. Make it bigger by the strap in the right corner, and we can go along with this. Maybe the application have a problem with full screen.
So now you are able to see my screen and changing the slides.
No, no.
Just wait for a minute. एक चीज़ जो है मैं इसको शेयर किया है लेकिन वो आगे ग्रीन जब मैं इसको करता हूँ तो विंडो ये मेरा है इसको शेयर करूँ हम इसको वो इधर क्यों से चली जाती है ये इधर आती है टीवी Salam. Saleh, are you
Yes,
there?
uh,
Can you move the slides?
I run the slides.
Yeah, I have made
Yeah,
you
no
the.
problem.
Hmm? Oh, he
Yeah,
will share his own slide. Hmm? Do you have the PowerPoint or not, Saleh? Yes, yes.
yeah, I have.
Then
Send
you
me a
can.
slide.
Then you stop sharing. Can you control this or no? No, he cannot. No, that's the thing. No, he cannot control. Reclaim. He cannot control. He cannot control. So I will stop sharing and you start sharing the file which I have sent to you. Okay?
Yeah, no problem. On it.
Okay. Yes, Saleh, you can share the slides with us.
You send it?
Hmm? Yeah, the first lecture of the pain.
Did you send it in WhatsApp?
Yeah, I sent it in the WhatsApp before to you.
Oh, it's the 3 and 4. Yes, yes. I have
Yes,
it.
it is number three, lecture number three, pain number one. Okay.
Okay. يلا should
All right. Uh, Now you can go to slide number four. Yeah, we covered this, that these are different phases. Next slide, five. All right. As I mentioned that pain receptors are uh, present all over the body. Basically, they are free nerve endings. And in this diagram, you can see that you have got different types of receptors. We will not talk about those, just the one which is in the red box. These are the free nerve endings. And these are most abundant of all somatic receptors, at least 9 times more than the touch receptors and around 15 times more than the thermoreceptors. Okay. All right. What do these receptors have? They contain certain ion channels. As you know, electrical signal will always be through a movement of ions. So if you have got some positive ions moving in, whether these are sodium ions or hydrogen ions or some other ions, so you will have creation of an electrical signal. So these substances which can open the ion channels in pain receptors, these are called algogenic substances. Algogenic. Okay. So we sometimes ask you about algogenic. So you should understand the name. One very important algogenic substance is histamine. Another one is bradykinin. Histamine comes from mast cells. These cells are present wherever there is inflammation, and bradykinin comes from plasma. So it is a protein there in plasma, kallikrein, from where bradykinin comes. Then also prostaglandins, which are fatty acid derivatives and that are released from cell membrane. Whenever the cell membrane gets damaged, it will release prostaglandins. Okay, so these prostaglandins will also act as pain-producing substance. Next slide, please. So we do have different types of pain receptors in the skin and in the mucous membranes. One type of receptors, they respond to mechanical damage, such as cutting, crushing, pinching, etc. So these are called mechanical nociceptors. So they are not mechanical receptors, they are mechanical pain receptors, mechanical nociceptors. Okay. The other group would be thermal nociceptors, which respond to extremes of temperature. Again, they are not thermoreceptors. They do not tell us about hot or cold, but they respond to very hot or very cold temperatures where these temperatures cause tissue damage. So these are thermal nociceptors. And the third group is polymodal nociceptors. These respond to a number of types of stimuli, including mechanical, including thermal, and then there are so many chemicals which produce activity in these free nerve endings. So they are called polymodal or polymodal nociceptors. Okay, is it clear? So mechanical and thermal, and the third one is polymodal. Next, please.
Clear.
Next slide. So this is just explanation of what I have just mentioned. So whenever there is tissue damage, a number of chemicals are released. Now some of these chemicals, they are the actual activators of these receptors. So we call them activators, such as potassium, serotonin, bradykinin, and histamine. They are the ones which actually activate the nociceptors. And their sources, as written here, damaged cells, platelets, plasma, kininogens, histamine mast cells. These two were discussed in the last slide also. Now, the other group is called sensitizers, meaning they sensitize the receptors to activators, making them more sensitive. So, even a smaller amount of activator will produce more response. So to give you an example, prostaglandins, they are the very important ones in group of sensitizers. Okay. If prostaglandin is present, maybe two potassium ions can produce a signal in nociceptor. If prostaglandin is not present, maybe you need 20 potassium ions to produce a signal. Okay. Is it clear?
Clear.
Yeah. So in sensitizer group, prostaglandins are the most important, but leukotrienes and substance P, which substance P is a neurotransmitter which is released in at the first synapse. The first order neuron secretes substance P for second order neuron activity. So these are also amongst the sensitizers. Next, please. So just to give you a broader idea about bradykinin, which is the most important pain-producing substance. Normally, it is present as an inactive substance, gets activated by enzymes that are released in the extracellular fluid from damaged tissues. Now, this bradykinin, this is responsible for a slow aching pain that you can feel when polymodal nociceptors are stimulated. So you need to remember this thing that bradykinin is the one which is most commonly activating the polymodal nociceptors. And this bradykinin, because it is present in the extracellular fluid, it remains there in the extracellular fluid for quite a long time. So for as long as bradykinin is present, the person keeps on feeling the pain. So though the initial cause of the damage is removed, but because bradykinin is still present, the person will keep on feeling the pain. Okay?
Okay.
Okay, next please. Now, substance P, as I just mentioned, this is released from the primary afferent pain interval. So it is released at the first synapse in the pain pathway. So it is activating the ascending pain pathway, and it helps in transmitting the nociceptive signals to higher levels for further processing. Now, a number of pain relief creams which are present over the counter also in the pharmacy, they have got a substance which is called capsaicin. Now, this capsaicin is basically an ingredient in chili peppers. red hot chilies or green chilies. So this capsaicin is a ingredient of normally occurring chilies. So what is the function of this capsaicin? It can actually cause the massive release of substance P from local nerve endings. So if the substance P is depleted, it is exhausted from the nerve terminals, so the signal will not go forward. So you just keep in mind the uh, overall mechanism that capsaicin depletes the nerve endings from substance P. So the onward transmission of pain is blocked. So the pain is relieved. If the signal does not go forward, the brain will not have any activity related to this pain.
Deplete means
Deplete
block.
means exhausts, finishes.
Oh, okay. Finish.
Okay.
Yes.
So next, please. So what we have discussed a short while ago that you can have two types of pain. One is fast pain and another slow pain. Very simple to remember. Fast pain will be transmitted by myelinated fibers. And the myelinated here are the very thinly myelinated A delta type of fibers, where the velocity of conduction is around 6 to 30 meters per second. And then the slow pain, which is transmitted by C type of fibers, which are unmyelinated fibers, okay, which are much slower as compared to A delta fibers. Now, if there is tissue damage at certain place, initially you feel a pain, which is fast pain. So that is transmitted through A delta fibers. And then the other type of pain, which is transmitted by usually polymodal nociceptors, this is transmitted by C type of fibers. So that's why you have got an initial sharp pain if you get a cut on your finger or hand, transmitted by A delta fibers, and then you have got a persistent slow pain which is transmitted by C type of fibers. So both type of fibers are coming from the same area but from different receptors. Fine?
Yes.
Okay.
Next.
You can go forward. So this is just to remind you that A delta fibers, this is the, if you go from the right side, second last on the right, the last one is the C fiber. Okay. This is unmyelinated. And then on to left of that is the A delta fibers, which is the fibers used by the fast pain. So this is the difference. And this is thinly myelinated fibers, and the C type of fibers are the unmyelinated fibers. And on the left side, it is clearly written that A delta fibers basically are coming from the mechanical nociceptors and thermal nociceptors, whereas the C fibers are coming from polymodal nociceptors, different chemicals. No, you did not mark it properly. Thermal nociceptors and mechanical nociceptors, which are related to pain in A delta fibers. You put it one line up, yes, and you delete the upper one.
Okay.
Okay, next slide, please. So the same thing again, but now where does it go in the brain? So fast pain or sharp pain that is transmitted by A delta fibers, it will of course enter the spinal cord and it will travel up, and then it ultimately terminates on second neurons which are present within the spinal cord. Okay, here the neurotransmitter is glutamate. So for fast pain, The neurotransmitter is glutamate. Then the second order neurons, where the cell body of this neuron will be in the spinal cord. Okay. So second order neurons, they cross over to the opposite side and that will go up till thalamus. Okay. And from thalamus, the third order neuron will go to cerebral cortex, the pain areas in the brain. You can actually localize the fast pain quite well. Hmm? But when will you be able to localize it well? When the surrounding touch receptors are also stimulated. So tactile means touch. So whenever there is a mechanical damage, so for example, if there is a cut in the finger, the mechanoreceptors at that site are also stimulated, of course, because they have felt this touch, sharp touch. And then because there is tissue damage, so mechanical nociceptors are also stimulated. So you can very well localize from where this pain is coming. All right. So fast pain, glutamate is the excitatory transmitter, and it is well localized with the help of mechanical receptors, mechanical touch receptors. Okay, next. The other one is the slow pain. You remember that slow pain is coming from polymodal nociceptors. It has got a character which is sort of burning and it is diffuse, aching type of pain, transmitted by C fibers. Again, similar to what we have seen in the last slide, the second order neuron, it will cross to the opposite side, okay, in the spinal cord. Now, the neurotransmitter here is mostly substance P, okay. So, in the slow pathway, substance P, and here the ability to localize the pain is not very good. So, poor localization. Here it is, poor localization. You will just say that I am having pain in my hand or in my arm or in my leg like this. You are not able to exactly pinpoint from where the pain is coming. So this is the difference between fast and slow pain also. Next, please. So these are differences. Sometimes we give you a short answer question asking you to differentiate between fast pain and slow pain. So you will have to just put these things into a table form. Just a quick summary that fast pain occurs on stimulation of mechanical and thermal nociceptors, whereas slow pain is produced because of polymodal nociceptors by substances such as bradykinin, capsaicin. Now, the transmission is by A delta fibers in fast pain, slow pain by C. And of course, fast pain occurs first and slow pain occurs second, slower. Okay. So you can just quickly review this. Next, please. Now the pain can be modulated also, meaning the character of the pain can be changed. How? Body itself, Allah subhanahu wa ta'ala has given it certain properties. There can be some suppression, attenuation mean decreasing of pain transmission and perception because of certain mechanisms. Number one is inhibition of pain pathway by stimulation of mechanosensory pathway. This is what is called gate control theory. And the other one that is present is descending pathway present in the body, which is called endogenous analgesia system. And we will describe them now in the next slide. Next, please. So, what is gate control theory? What you see in the left panel next to where it is written stimulation of type A beta fibers from peripheral tactile receptors can decrease transmission of pain. So what you see here, if you start from the bottom, nociceptor is written here, and it is a C fiber that is taking the pain signal. You go straight up, The second order neuron is present in the spinal cord, and then it goes up also to the thalamus and then to the cerebral cortex. What you see here is a collateral branch coming from this C fiber. This is a branch. And what this branch is doing, hmm, this branch is basically synapsing with a interneuron. And this interneuron, if you are on the left panel, please. Yeah. This is a inhibitory interneuron, which means that when this interneuron is activated, it will inhibit the second order neuron. So this is the function of this inhibitory interneuron. Under normal circumstances, what is happening? Whenever nociceptor is stimulated, the signal inhibits this inhibitory interneuron. Again, remain on the left panel, please.
Sorry,
Yeah.
sorry.
Yes. So, whenever the tissue damage occurs, the signal goes, and this inhibitory interneuron is inhibited. Means it will not inhibit the second neuron, and the pain signal will go up. Clear?
Clear.
Yeah. Now, if you do a massage, now come to the right panel. So this is now from skin mechanoreceptors. If you stimulate these mechanoreceptors, what do they do? You will feel the touch of the massage, of course, but in addition, a branch of this A delta fiber or A beta fiber, it stimulates this inhibitory interneuron. So if inhibitory interneuron is stimulated, it will inhibit the second order neuron in the pain pathway. So the signal that was going for the pain, it becomes weaker and weaker and weaker. So person gets relief. Person gets relief. So this is the theory that is put forward for massage, for acupuncture, for electrical stimulation, all those gadgets or processes that are used to reduce the pain. They are basically thought to be working on this mechanism that they stimulate an inhibitory interneuron which inhibits the second order neuron in pain pathway. Okay?
Okay.
Okay. Next slide, please. Next, please. Yeah. So this is what I have just mentioned. Huh? So most of these mechanism, basically, if the physiological term we use is lateral inhibition. So touch pathway is inhibiting the pain pathway. This is lateral inhibition. So as I mentioned, this is thought to be the mechanism of action of massage, certain creams, and then this electrical stimulation of skin, tense, transcutaneous electrical nerve stimulation. Next, please. Now the other mechanism, which is called the endogenous analgesia system. What does it mean? That some higher influences, maybe you are in a battlefield, so the thinking is to do something extraordinary. So what happens? Those higher influences, they can stimulate an area in the midbrain, which is called periaqueductal gray matter. Now, if this periaqueductal gray matter is stimulated, all right. So periaqueductal gray matter would further stimulate certain areas in the medulla, such as nucleus raphe magnus, locus coeruleus, and medullary reticular formation. So These areas, when they are stimulated, they are using certain neurotransmitters such as norepinephrine, dopamine, serotonin. So they release these neurotransmitters. And ultimately, what do? They stimulate certain inhibitory interneurons in the spinal cord. So what I have just mentioned is the periaqueductal gray matter. It stimulates certain areas in the medulla, and these areas in the medulla stimulate inhibitory interneurons in the spinal cord. These inhibitory interneurons that are present in the spinal cord, they release opioid, which is called enkephalin. If you go to the next slide, please. So yeah, this is diagrammatic. So periaqueductal gray matter is present in the midbrain. So something message is coming from the higher center. So this periaqueductal gray matter is stimulating certain nuclei. Here the nucleus we are showing is nucleus raphe NRM in the medulla. And this NRM further stimulates an inhibitory interneuron in the spinal cord. Okay, so this is enlarged here at the bottom. So what do you see? There is an interneuron. So this is the inhibitory interneuron that is stimulated by this pathway. And this interneuron releases a neurotransmitter which is called enkephalin. Okay? Yes. So this diagram further gives you the idea. This is the inhibitory interneuron. Once it releases the enkephalin, what does enkephalin do? It goes and binds with the presynaptic membrane of the first order neuron and stops the release of substance P. It stops the release of substance P. So if substance P is not released, the second order neuron, of course, will not get any message. So no perception of pain. Some of this enkephalin also acts on the second order neuron, this dorsal horn excitatory neurons, and make it less excitable. So this is how the endogenous analgesia system works. So we hear that a person, he was fighting and his hand was cut, and even the bleeding was there and hand was cut. He kept on fighting till he reached his goal. Maybe he won the battle, or he wanted to reach a certain place, whatever. So the higher influences in the cerebral cortex, his thinking, his emotions, they are affecting and producing ultimately an activity through this inhibitory interneuron where the pain pathway is suppressed and the person will not feel pain as long as he is in that particular situation emotionally and physically. Once that event is finished, this stimulation will also finish and he will start having release of substance P and he will start feeling the pain. Is it clear?
A little bit,
Yes.
yeah.
So now you have to differentiate two things. One is endogenous, which is from within the body. This is using this system, periaqueductal gray matter, stimulating the medulla stimulating the inhibitory interneuron in the spinal cord, and then this is suppressing substance P production, release. The other one is external, where we do the massage from outside, or we use acupuncture, or we use certain creams, etc., whatever. So
Left
this is
one
the
is
gate
internal,
control. This is from
right one is
external.
external.
Okay.
Yeah.
So as physiotherapists, you will be using this mechanism, the gate control theory.
Okay.
Okay. Yes. So some of the endogenous systems that are available in the body, These are called the neurotransmitters that are used are beta endorphin, metenkephalin, leuenkephalin, dynorphin. And as I mentioned, their function is pain suppression during stress or during certain periods of heightened cerebral activity. Next, please. Now, what happens further up? Spinal and higher level processing of the pain. So, what has been experimentally seen is that person can perceive pain at thalamic level, but this perception is not accurate. That is, the if the cerebral cortex is not involved, then the animal studies have shown that even at thalamic level, the animal can exhibit certain behavior indicating that it is feeling the pain. But the pain localization and the intensity of pain cannot be determined by thalamus. These two things, that is, the localization of pain and how intense is the pain, this is the function of cerebral cortex. Okay.
Okay.
Now, as we discussed in the very beginning, that the pain sensation is accompanied by certain behavioral and emotional responses also. So these responses, they occur because of involvement of hypothalamus and limbic system, that is in the a deep system in the cerebral cortex. Also, the person feels very much uncomfortable and alert if the pain is there. So this alertness occurs at the level of reticular formation. So these things are given in the next slide. Yes. So this diagram gives you, if we just go to the dorsal horn interneuron and we start going up towards thalamus. So you see a branch going to reticular formation. This is to keep you alert. The person cannot go to sleep if he is in great pain. And from reticular formation, the signals are also going to hypothalamus and limbic system. Even from a higher level, from thalamus, the signals are going to hypothalamus and limbic system, and these two are responsible for your behavioral and emotional responses, meaning the person starts crying, producing certain sounds like ah, oh, like and the facial expressions are also there of very pain, having great pain. So we are having a signal going in multiple directions. One is for to the somatosensory cortex for actual perception of the pain and localization of the pain. The other ones for behavioral and emotional responses, and the third one for alertness. Okay, next please. So with this we finish the lecture number one. Uh, Now what should we do? We start the second session at 1:30.
Yeah.
Hmm? All right, because now it is Salah time also, and we have got another meeting immediately after Salah. So inshallah,
Okay.
hopefully by 1:30, I'll send you another link, and then we can do that. Okay?
Okay, doctor.
Okay, shukran. Assalamu alaikum.
Upon